Okay, I'll get started here with the introduction. So it's my great pleasure to introduce our second keynote speaker, Anshul Kundaje. He's an associate professor of genetics and computer science at Stanford. His research focuses on computational regulatory genomics, where he develops statistical and machine learning methods that analyze large-scale functional genomics data. Anshul completed his PhD in computer science at Columbia in 2008, and following his doctorate, he pursued a postdoc at Stanford from 2008 to 2012, and then he was also a research scientist at the Broad and MIT from 2012 to 2014. During this time and throughout the rest of his career, he's played a leading role in the integrative analysis efforts of several major consortium, including serving as a lead analyst for ENCODE and a lead analyst for the Roadmap Epigenome project. His contributions have been widely recognized with several honors, such as the NIH Director's New Initiative Award and the Alfred P. Sloan Foundation Research Fellowship. Very relevant to this conference, Anshul has integrated cloud computing into both his research and his teaching to enhance the access and accessibility of large-scale genomic data. At Stanford, he teaches cloud computing for biology and healthcare, which I just was reading about. A course that educates students on leveraging cloud platforms for computational biology and healthcare applications. In his research, he utilizes cloud computing to manage and analyze extensive genomic data sets. His lab develops machine learning methods for integrative analysis of functional genomics data, which often requires very substantial compute resources and GPU resources. Cloud platforms provide the scalability and processing power necessary for these analyses. By incorporating cloud computing into both his education and research, I like to think that he advances the entire field of genomics, promoting more efficient data analysis and fostering innovation in biomedical research quite broadly. It's my great pleasure to introduce you. We're very sorry you couldn't be here in person, but we're glad that you can at least join this way to tell us about your research. Thank you, Anshul.
Thanks, Mike. Hope you can hear me. Yeah?
Yes.
Great. Okay. Yeah, so firstly, my apologies for not being able to make it in person. I came down with something like the flu over the weekend. I didn't want to come infect everybody else. But I'm feeling pretty decent right now, so I'm happy to be able to at least present this talk and the tutorial later that we are going to be doing with my postdoc, Vivek, who's there. I'm going to focus on this topic of using machine learning models, specifically deep learning models, to understand non-coding DNA and use these models to decipher genetic variation. So let's just give a brief introduction to the topic. So we're going to focus on transcriptional regulation. What I mean by that is, as you know, genes encode for proteins, and specific repertoires of genes are expressed in specific cell types, giving rise to the entire diversity of function and morphology that we see across the diverse cell types and tissues in the body. So to regulate expression of genes, evolution has produced these regulatory elements, as shown right here, like promoters and enhancers. These are short sequence elements that actually control gene expression. The way they do this is the sequences of these elements encode complex syntax of sequence motifs, as shown right here. These sequence motifs are recognized by DNA binding proteins called transcription factors. These transcription factors typically bind DNA in a combinatorial manner and give rise to complex activation repression effects on their target genes. So we're going to focus really on this question of trying to decipher the encoding of this sort of regulatory information in the non-coding DNA sequence of these regulatory elements. And first, we have to try to identify where these elements are. And so there are a variety of different profiling approaches, genome-wide high-throughput sequencing-based approaches. For example, you can use transcription factor ChIP-seq experiments. These will allow you to profile binding sites or occupancy profiles of individual proteins. Each row here is a different protein. And this is some locus in the genome. This is all ENCODE data, ChIP-seq data in one cell line, K562. Each row, again, as I mentioned, is a separate protein. And you can get these kind of beautiful genome-wide binding maps of proteins in specific cell types. But this requires a single experiment per protein in a cell type of interest. So if you were to try to cover the entire set of transcription factors in all possible cell types, this would essentially be infeasible for various reasons. So there are other approaches which don't measure binding landscapes of individual proteins, but give you genome-wide landscapes of regulatory DNA, typically regions that are bound by any protein. This includes assays like ATAC-seq and DNA-seq. ATAC-seq uses a TN5 transposase. It preferentially cleaves open chromatin, which is typically bound by transcription factors. DNA-seq uses a DNase-1 enzyme. And then there are single-cell versions of this like single-cell ATAC-seq. So the nice advantage of these assays is you can perform one experiment in a cell type of interest, and it gives you a genome-wide map of most regulatory elements that are open and accessible, but they don't tell you precisely which protein binds each of those elements. And so either way, as I mentioned, our goal is to take these kinds of experiments and try to learn models that can explain how the underlying sequence of these elements drives their activity in a cell type or tissue-specific manner. And these kinds of maps have now been produced on a massive scale. So large consortia, as well as other open efforts, have generated these kinds of terascale maps. On the x-axis, on this axis, you can see the whole genome, so 3 billion positions. At each position, you measure some kind of molecular readout through one of these experiments I mentioned: ChIP-seq, DNase, ATAC, RNA-seq, ProCap. And each of these experiments is often performed in a wide diversity of cell types, tissues, individual cells, and even spanning multiple individuals, so different genetic backgrounds. And of course, this map is not complete. It's quite incomplete, but it's still a very large reference map. Think of it like the reference, just as we had the reference genome about in 2003 and newer versions of it now. Think of this as sort of a reference regulatory map of the human genome. And so these maps are quite dynamic, as I'm showing you right here. If you look at how a particular promoter of a gene or enhancer of a gene is changing its activity across different cell types, each row is a different cell type. You can see, for example, this element is active in these collection of cell types and inactive in these cell types. And so there's lots of dynamics across the different cell types. And this regulatory dynamics gives rise to differences in transcriptional programs that define cell state and cell types. And so it's very interesting to try to understand how the same sequence is sort of driving differential activity in different cell types, right? And so that's where we come in. We try to train machine learning models on this type of data, genome-wide profiles of activity. And we use these models to interpret them and decode the underlying sequence, and then use them in a counterfactual manner to predict effects of perturbations. For example, genetic variants, mutations, or we can do even synthetic design or synthetic perturbations of DNA sequences and try to predict their activity. So I'm going to talk about a class of machine learning models. These are what we call base resolution deep learning models. And I'll explain what this means. Very briefly, what this is trying to do is for any given experiment, let's say you're measuring some ChIP-seq profile or ATAC-seq profile across the genome, the goal is to learn how sequence drives that activity. So this model is trained to learn a mapping from sequence across the genome to activity across the genome at single base pair resolution. So what we're doing here really is we're taking the FASTQ files that come out of the sequencer, we map the reads to the genome, and we simply compute the coverage at every position. So there's no processing beyond that. Okay, it's very simple. We're just going to count reads across each position in the genome. Sometimes it's stranded, sometimes it's unstranded. Either way, it's very raw data, base pair resolution coverage. And the neural network that we're going to train, and I'll explain what these neural networks do, but the neural network that we're going to train is, think of it like a text-to-speech converter. The text is the DNA sequence, and the speech is like this sequencing experiment signal. And the model, the neural network, is trying to learn to take the underlying sequence and transform it through these various operations, nonlinear operations, to predict or map it to the profiles at single base pair resolution. And we've trained many such, we've built many such models starting off in 2021. The first was this BPNet model, and I'll talk about that quite a bit today. And Vivek, who's there in the audience and is going to run the tutorials, we'll actually show you how to train these kinds of models on AnVIL. But these are models trained to map sequence to TF binding landscapes. So ChIP-seq, ChIP-exo, cut-and-run experiments. We've recently also built versions of this model for chromatin accessibility. That is DNA-seq and attack-seq and single-cell attack-seq experiments. I'll talk about that today as well. What I won't talk about is other versions of these. So for example, we've trained similar models for nascent transcription, steady-state transcription from ProCap and CAGE and Rampage experiments. And coming soon, we have similar models for reporter assays as well. So one aspect of our models is that they are very efficient. They're very quick and easy to train. I'll give you some cost numbers and so forth in the coming slides. So we've sort of designed them to be very cheap and also highly performant. And I'll show you some very powerful applications of these models and comparisons to much, much bigger models. And the key idea is we train one model for every experiment, okay? So if you perform an experiment, the goal of this model training is to encapsulate that model, encapsulate that experiment to a model which you can explain how that experimental profile that's produced across the genome is driven by sequence. And our models are quite local. That is, they try to map relatively local regions of sequence, so about 2,000 base pairs of the genome to corresponding profiles. Now, of course, there are many long-range interactions in the genome. So, you can have an enhancer 100 kilobases away affecting expression of a gene very far away from the promoter. But currently, our models are just focusing on what is the effect of local sequence on activity that you see locally, okay? So they can't explain a lot of long-range effects, but I'll show you how they actually can often perform as well or better than current generation long-range models, okay? So we're going to focus on kind of these small local models. You can ask me in Q&A why we focus on these, but that's what we'll talk about today, okay? Now, the way we train these models is we train them across the genome. So we tile the genome into bins, and we walk across the model, across the genome. It has sequence and the profile, and it learns this sort of mapping from sequence to profile. We train the model on a subset of chromosomes, and then we test the model's predictions on held out chromosomes not seen in training. And that's important because you want to understand how well the model can do on sequences it has not seen in training. It's very easy for a neural network or any other machine learning model to simply memorize the data. So it's very important to evaluate its performance on sequences not seen in training. So that's the procedure we use, okay? So once we train this model, what are we going to do with it? Now, the experiment is already available, right? So there's no really use in like predicting some, the profile on a held out chromosome because you already have the data. But that's just a way to evaluate the performance of the model. And once you know you have a good model, you can do many interesting things with it. So here's one thing you can do with it. Let's say there's some regulatory element in the genome, and the model is now trained and is able to accurately predict activity of that element. You can now ask the model, how are you making this prediction? Can you tell me which bases in the sequence underlying that element are driving activity? So we've built a series of tools shown here, DeepLift, FAST, and FastISM. Usually what these interpretation methods do is they allow you to interpret individual sequences through the lens of the model. So what the model does is takes the sequence, makes a prediction, and what these methods do is they take the prediction and they then deconvolve how individual neurons in the neural network are actually helping make those predictions. So we backpropagate the prediction from the output all the way to the input, and you get a contribution score per base. And that contribution score tells you literally the contribution of that base to the prediction. And the way these contribution scores work is they're a simple additive decomposition of the prediction of the model, okay? So you take the prediction of the model, you usually subtract it from some reference prediction, some kind of, let's say you take a shuffled version of the sequence and you push it through the model, that's kind of like a baseline null prediction. So you take the prediction of the actual sequence, you take that baseline prediction, you compute the difference, and that difference is what you're trying to explain with this method called DeepLift. and that gives you a contribution score per base. If you add up those contribution scores, they equal the difference of the prediction of the model on the actual sequence minus the prediction of the model on the reference shuffle sequence. Okay, so think of it like it's predicting fold changes of the prediction of the model with respect to some reference. Okay, but it's nice interpretation because once you add up these scores, they literally give you the prediction of the model. So it's a very simple additive decomposition. Even though the neural network is a complex nonlinear model, the contribution scores allow you to get a very simple first order interpretation of how individual base pairs in the sequence drives activity. So this is useful if you want to interpret individual sequences, right? These could be genomic sequences or synthetic sequences, whatever it is. You can push them to the model can predict activity, and you can use these methods to infer the contributions of individual base pairs. Now, what you might want to do is actually take a collection of sequences. Here I'm showing you 3 sequences, but let's say you have tens of thousands of binding sites of a protein, and the model has correctly predicted the profiles for all of those sequences. You can apply this sort of interpretation method, deep lift, as I showed you before, for each of these sequences through the lens of that model, and the model will tell you which bases in each of those sequences are driving activity of those sequences. Now that's a lot of patterns, right, that the model is showing. So for example, the model here is showing this sequence, and on a different promoter enhancer, it's showing a different set of sequences. So that's a lot of sequences to interpret. So you might want to summarize these into kind of a non-redundant set of patterns or motifs that the model has learned over all the sequences. So TF-Modisco is a method that does that. What it does is it takes these tens of thousands of sequences, we interpret them using the model with deep lift. We get these contribution scores, which tell us which bases are important. We throw away the base pairs that the model claims are not important in each of the sequences. So we end up with these little seclets. Seclets are short sequences of high importance. And then we run a clustering algorithm. That's TF-Modisco. It's sort of a community detection method. And it basically what it does is compute similarities between all these little seclets after aligning them, does this very quickly, and then clusters them into groups of sequences that are very similar. And then we align and average them out to produce these kind of lexicon of predictive patterns that are seen across a large family of sequences. So you can kind of pick and select whatever sequences you're interested in, and Modisco will summarize all the predictive patterns in those sequences. Okay. Now you can do other interesting things too, beyond the lexicon, which is just individual words and motifs that the model has learned. You may be interested in more complex syntax. So one example of syntax would be spacing, right? So maybe you're interested in how is the relationship, how does the spacing between two motifs influence the activity predicted by the model? So we can perform various kinds of in silico perturbation experiments. For example, we can design synthetic reporters where we take artificial synthetic sequences. We can insert motifs. We create a library of sequences where you insert 2 motifs and all you do is change spacing. And then you use a model effectively like a reporter experiment, but in silico, right? You push all that entire library of sequences through the model, and the model will make predictions for how small changes in spacing between these motifs affect activity. And so you can use a model as an oracle. You can probe it and try to understand complex aspects of the underlying syntax beyond individual motifs, right? You can mutate bases within motifs to understand effects of affinity. You can change spacing, you can change orientation, you can change density. You can do lots of interesting experiments. You can also do the equivalent of CRISPR experiments. You can take actual genomic sequences and you can do combinatorial mutations. You can mutate 1 motif or 2 motifs or individual base pairs, whatever you like, right? And of course, then an interesting application of this for natural perturbations, genetic variants, is in fact variant effect prediction. So you can use a model to actually screen for various kinds of non-coding variants, common, rare, SNV, indels, doesn't really matter. As long as it's a perturbation to the sequence, you can take a reference sequence, you can push it through the model, you can make a mutation coming from, for example, a natural variant that you see in some patient or in an individual. You can take the reference allele, the alternate allele, push both sequences through the model, look at the delta of the predictions, that gives you kind of a predicted effect size, right? So these are all the applications you can accomplish with the models. So that's the toolkit. So now let me walk you through some actual examples of how we use these models in practice and what we can do with them on a large scale. And I'll start with BPNet. So this is a bit more detailed dive into what the BPNet model looks like. As I mentioned, it is a local sequence model. So it tries to map 2000 base pairs of sequence tiled across the genome. You can just take any 2000 base pair sequence, and it tries to map that 2000 base pair sequence to base pair resolution coverage profiles. Okay? It's literally predicting read counts at single base pair resolution. And the neural network architecture is very simple. It's not one of the cutting edge architectures. It's a very simple convolutional neural network. These convolutional neurons, they are pattern detectors. They learn motif-like patterns, and they scan the sequence with these patterns. And then these additional layers of convolutional filters. So the model has like 9 layers in this case. Each layer builds on the patterns of the previous layer and learns more complex patterns. And so as you add more layers, it can learn not just individual motifs, but much more complex syntax, density effects, affinity effects, effects of spacing, orientation, higher order interactions, and so forth. And the last layer of the model, so you think of it like, starting with sequence, and then the neural network is processing that sequence through these transformations. And then the last layer of the model is trying to emit or predict the profiles at base pair resolution. And it does this in 2 ways. There are 2 outputs actually for the model. One output is the total coverage over the entire 2000 base pairs, right? So that's one number that just gives you the total coverage. We actually predict the log of the total coverage. And then the second component of the model takes the total coverage, the total number of reads, and learns to distribute them across each of the base pairs in the sequence. So we kind of have these 2 complementary tasks trying to predict total activity of the sequence, and then the base pair resolution distribution of that activity across each base pair, okay? And so let me show you what this does. This is a TF ChIP-seq data. And here we have 2 separate data sets, okay? This is one region in the genome. It's about 200 base pairs. And we're looking at ChIP-seq data. Both these experiments are trying to measure the same binding of the same transcription factor, E2F6, okay? So they're supposed to capture the exact same data. But, as you know, these experiments often have, highly differing data quality. So the one on the top is actually a high quality experiment, and the one on the bottom is a much lower quality experiment. And you can see the y-axes are very different. This data set has, the maximum scale is 4. So this position has 4 reads. You can see this has 1 read, and you can see there are 1, 2, 3, 4, 5, 6. There are just 6 reads in this region for the lower quality data set, but there are, like 45 or so reads for this data set, right? So this data set is called as a peak, as a binding event in the high quality data set, but in the low quality data set, it's not at all called as a peak. It's just basically like random background. I just want to show you this because you can clearly see how different these experiments are. And actually, if you just look at the number of peaks or binding events called across the entire genome for the 2 experiments, this data set has about 4 times as many peaks as this one, okay? There's a huge difference in data quality, and they look completely different, even though they're supposed to measure the exact same thing. So now what we can do is train separate models on each of these data sets. And the way we've designed, the loss functions that we use to train these models is to really adapt to the sparsity of the data. So what these models can do is once you train them, they make predictions at base pair resolution. Even at positions where you don't have signal, the models can predict the expected value, right? Because the model is basically borrowing power across all the sequences in the genome and learning how sequence maps to activity. So then even at positions where you don't observe reads because of the sparsity of the data, the model can still make a prediction of the expected value, right? From the model. And when you do that, you get these beautiful predictions. So this is the exact same, again, it's 2 separate models that have never seen each other. They're trained on each of these separate data sets. And when you train those models and you now make predictions on that exact same region, you can see the model predicts this really amazing profile. You can see it's like here's one strand, here's the other strand. You can see this beautiful symmetry. The binding site is right in the middle right here. And even for the really lower quality replicate, where you have 5 reads, the model imputes the missing signal and looks very similar to what you see for the high quality data set. And not just that, but now if you use the interpretation method deep lift to explain this profile that the model predicts from the 2 different models, you can see that the models make the exact same inference of what is driving the prediction as well. And if you look at these motifs that are being highlighted by the contribution scores from both the models, it makes a lot of sense. This motif right here is the E2F6 motif, the motif of this protein itself. And this motif is the motif of MAX, another transcription factor. And these 2 motifs together are collaborating. So what this is telling you is that E2F6 is binding its motif, but there's another protein MAX binding its motif, and the 2 motifs are collaborating to drive activity exactly the same way in the 2 different data sets. So this is quite remarkable. You can see the dramatic improvement in predicted improvement in concordance of the profiles after prediction, and an improvement in concordance of the interpretation itself. If you looked at these observed data sets, you would say, oh, there's no binding here, and there's binding in this one. But here you can clearly see both models predict binding, and they give the same explanation. Now we can do this genome-wide. We can not just do this for one region in the genome, but across all the peaks. And on the x-axis, I'm showing you the Jensen-Shannon distance, which is a measure of distance between these 2 profiles and these 2 profiles. So think of it as very low values indicate very similar profiles. Very high values close to 1 indicate very different profiles. So you want these to be as low as possible. Now, if you look at the distribution of these distances for the observed data, that is, if you take the high quality replicate and the low quality data set and you compare the distributions across the base pairs for all the peaks in the genome from the observed data as measured by the experiment, you see very poor concordance, right? They literally look like completely different. They're very close to 1, but the purple bar, the purple curve is showing you similarity or distance or distributions for the predicted profiles from the 2 models. And you can see this amazing dramatic improvement, right? Like really beautiful denoising from the model. And not just for this data set, but we have now thousands of data sets from ENCODE where the same protein has been chipped through to 2 different experiments. We compute this delta JSD, this improvement in data quality compared to the observed data. You want this number to be very large and positive. And you can see that across the board for hundreds of data sets, hundreds of pairs of data sets, you can see this really beautiful denoising. So the neural network, is able to not only improve data quality, but also give you explanations, helping you understand what in the sequence is driving activity. So this is a very powerful way of using the models to actually just improve the data quality itself. So what we can do is not just predict, individual sequence motifs. We can also look at the effects of, as I said, spacing, right, and syntax between motifs. So when we run this BPNet model on ChIP-seq data for the FOXA2 transcription factor, the model, similar to what I showed you before, it denoises the data. We can infer the base pair resolution contribution scores of motifs in individual binding sites. You can run Modisco to learn motifs across all the binding sites. And the model discovers these 3 motifs. These are the main 3 drivers of activity, of binding activity of FOXA2 in liver, okay? One of them is the FOXA2 motif itself, and the other is the motifs of 2 other transcription factors, HNF4 and CBP. And these motifs are often found nearby in the genome, just like here we see how E2F6 and MAX are, but they're found at different spacings. So you wanted to understand, are these motifs kind of acting independently, they're just randomly placed, or is there some kind of really interesting spatial relationship between these motifs? So what we do, actually, this is Vivek's analysis. What he does is we do the synthetic reporter experiment where we take kind of randomized sequences. These are just kind of neutral background sequences, shuffled. And then we insert the FOXA motif at one position. And then we insert the CBP motif at another position, and we change the spacing, okay, between those 2 motifs. So the only thing that's changing between these sequences is the spacing between the 2 motifs. They're randomized sequences, and they have the 2 motifs, but we're changing the spacing. And we're systematically measuring through the model, we're predicting the activity, the binding activity of FOXA2 as a function of spacing of these two motifs. So x-axis is spacing, y-axis is the fold change of predicted activity or simple additive effects. So you can predict the activity of FOXA by itself, predict the activity of CEBP by itself, predict the activity of FOXA plus CEBP, assuming they acted independently and additively. And then you can predict the effect of FOXA and CEBP together jointly in the sequence through the model, right? So trying to look at the epistatic effect, the super additive effect of having the two motifs together at specific spacings versus how they would act additively independently, right? And you can see this really interesting behavior. Here's the animation of what's happening. As we're changing the spacing between the two motifs, watch the signal, okay, of FOXA2. As CEBP gets closer, you see how the signal starts rising? This is amazing amplification of binding of FOXA to its own motif due to the presence of another motif nearby, but it only happens at specific spacings, right? So you can draw this graph of the change in binding, and you can see that as you drive the motifs closer together, you see this exponential rise in binding. And here's just sort of snapshots of this, right? You can do this for FOXA and HNF4 as well to understand their spacing relationship. And they also have a very interesting cooperative spacing relationship. It's different from CBP. You can see that HNF4 can have pretty strong effects even further away. But the maximum impact is actually higher for CBP than HNF4, right? And you can see kind of different kind of curves showing you kind of different effects of spacing of two different motifs, CBP and HNF4 with respect to FOX. So we can start doing this for all kinds of pairs of transcription factor motifs learned by the model for hundreds of transcription factors that we've trained these models on, right? And that's exactly what we've done. This is where AnVIL comes in. To do this at scale, we really required something that we can really automate and run very quickly. And like I said, we designed these models to be small and cheap and highly performant. And so you can see here we've trained now models on over 3,500 ChIP-seq datasets from ENCODE, all of them for which we have data. And we've generated a compendium of these models and sequence syntax annotations for all these datasets. We've created these AnVIL workflows. You'll play with some of these during the tutorial that comes next. But we've basically run this for literally thousands of datasets. And you can see the model performance is very stable, irrespective of how many peaks each dataset has. And just to give you an estimate of cost, I said these models are cheap. They really are. Training one such model on one of these cross-validation folds costs just $1.25, okay, on AnVIL. And for inference and interpretation, it's $1.75. So to really interpret the contribution scores of all the sequences in the genome to which the protein is bound, make these predictions of these kind of denoise profiles, and then learn motifs, all of that costs effectively less than about $5, right? So that's really cheap, and that's how you're able to really scale this and do this extremely effectively. And all of these models and all the interpretations and motifs and so forth are coming soon. They're not yet publicly available. They'll be publicly released in about a month, but they'll be at the ENCODE portal at these locations. So if you're interested in playing with these models, and Vivek will show you how you can use these in the tutorials coming up next, but these will all be available very soon, okay? So that's, BPNet for transcription factor binding. We've now recently, as I mentioned, also developed this new model called Chrome BPNet, which is a model that's really designed for ATAC-seq and DNA-seq experiments that measure chromatin accessibility. It's exactly the same as BPNet, okay? It's the same model. That's the beauty of this architecture. It's literally the same backbone. The only difference is that since ATAC-seq and DNA experiments capture binding sites of hundreds of proteins, whereas ChIP-seq experiments capture binding sites of a single protein, you need to learn many, many more sequence patterns, right, to explain chromatin accessibility than TF binding. So all we do is the model architecture is the same, but we increase the number of parameters. So the number of pattern detectors in each convolutional layer is now many-fold higher, okay? So BPNet models have about 64 pattern detectors per layer, and there are 9 layers. Chrome BPNet models have 512 pattern detectors per layer. Okay, so that's an order of magnitude increase in the number of layers. Still a very cheap model. You can train it about, by the way, the BPNet models you can train in about an hour or two on a reasonable consumer GPU, whereas the Chrome BPNet models take about 6 to 8 hours depending on, or 6 to 12 hours depending on what GPU you have. But none of these require like extremely expensive GPUs. So we can train them at scale and get a lot of value out of them. Okay. Now, these models also perform very accurately. As you can see here, the performance, comparing the observed coverage against predicted coverage over entire peak regions, you get pretty decent correlation, 0.7. It's lower than replicate concordance. That's expected because our models only are modeling local sequence. You really have to model much larger swaths of the genome if you really want to predict the counts, the total coverage very accurately. But I'll show you why this is still very powerful. So the coverage is quite decent, but the profile prediction, the base pair resolution profile prediction is very good. It's nearly as good as replicate concordance. This is concordance between replicates. This is concordance between predictions and observed data. And this is sort of baseline, what a random prediction would look like. And if you look at any region in the genome, you can actually see that. This is a single sequence, 200 base pairs. And this is the observed profile, coverage profile at base pair resolution. This is a prediction from the model. You can see it's really indistinguishable to the eye. It's extremely accurate, right? But there's a problem. This looks really good in prediction space. When we interpret the model, things look much noisier and dirtier than what we saw for BPNet, okay? This is, we know this region. I specifically show this region because it is a known binding site of the CTCF transcription factor. And why I like CTCF is because it has this massive motif right here. And it's a very clean motif, and it's very clear that should be driving the signal. But when we interpret the model now, trained model, the model is trained on ATAC-seq data, right? So we don't know which protein binds there, but we have ChIP-seq evidence that CTCF binds this location. That's why we expect that the CTCF motif should be driving activity, right? Chromatin accessibility. But we see that to some extent. You can clearly see the CTCF motif driving activity, but there's a lot of other spurious signal right here. And this spurious signal did not make sense to us at all. It's just kind of random bases popping up and down all over the place. It's not just in this region in the genome. We find it all over the genome. So we wondered what is going on, right? So that's where Modisco comes in. This deep lift allows us to look at individual sequences and sort of debug what's going on. But we wanted to see if this pattern is making sense across the genome. So when we run motif discovery across the entire genome, we see something terrifying. We see the model learning TF motifs, CTCF, RUNX, IRF1. This is all good, right? This is what we want the model to learn. But we also see these kind of weird CAG repeat patterns all over the place. And if you just count the number of motifs, right, driving these patterns, it's orders of magnitude more than the TF motifs. And if we try to investigate what this pattern is, we realize, in fact, it is not anything biological. Well, it's at least not biologically interesting from a causal perspective of understanding the biology of transcription factor binding and chromatin accessibility. Instead, this is the sequence preference of the enzyme used by the ATAC-seq experiment called TN5. So TN5 is this transposase that goes and tagments open chromatin. And unfortunately, TN5 is not a neutral enzyme. It has its own sequence preference. And the neural network is so powerful that it is able to predict the data extremely accurately. But as in other domains, where neural networks not only learn the underlying causal signal you're interested in, but many other biases, in this case, it's doing the same thing. It's learning not only the underlying transcription factor sequence biology, but it's also learning the enzyme bias itself. And the enzyme bias is completely dominating the model. Okay? This is really bad. So how do we, first of all, two things to mention here. This shows the importance of model interpretation. You should not stop at simply comparing model performance, because when you look at the predictions of the model, you can feel very happy. You can feel like, oh, wow, my model is amazing. It's doing fantastic. But then it's very important to interpret the model, because that's when you start realizing, oh, wow, okay, the model is not just learning what I wanted to learn. It's learning all these other spurious biases, right? Helps you also debug the experiment, debug the model. Now we have to figure out a way to correct this bias. We can't live with this bias and go ahead with our life. We need to correct the bias. So luckily what happens is the human genome is very vast, and only 1% of the genome actually shows high signal in ataxic experiments, right? So that's great because the rest of the genome is like a control experiment. It's basically just background where no transcription factors are actively binding to drive accessibility. And so it's kind of a neutral control, right? So if the enzyme had no bias, then in the background regions of chromatin, you would just see random distribution of reads, right? So what we can do is, rather than training this model on peak regions, we train a model, the same BPNet model, the Chrome BPNet model. We train a neural network, not on peak regions where you're hoping to learn transcription factor biology, but rather we train it on the background. This might sound really dumb because what are you doing training it on background? Well, the idea is the following, that if there is no bias, right, then reads would be randomly distributed in background. And if you train a neural network trying to learn to predict that background from sequence, it would fail miserably, right? Because the profiles are literally random. There's no prediction capacity. But if the model is able to make very accurate predictions in background, it means two things. Firstly, it means that somehow you can predict background profiles from sequence, indicating that there's a very strong sequence bias. And then you would hope that this model would have learned that sequence bias extremely accurately. And that's exactly what we see. The model can predict the background extremely accurately. And when we interpret this background model, it learns 9 motifs. And those 9 motifs are exactly the CAG patterns, different variants of them. But it's basically learning the best possible model for TN5 bias. Okay, so now this is great because we have a very good model of TN5 bias. Then we have this horrible confounded model that has mixed the TN5 bias with TF biology. So we can train now a new model that separates the bias from the underlying latent signal that we want to learn. How do we do that? Now we go back to fitting models in peak regions, again, the profiles and the counts. But rather than fitting one model, we fit 2 models in two phases. Remember, we've trained this bias model on the background chromatin. So we first try to use the bias model to predict bias in the peak regions, right? The bias model is trained on background regions, but now once it's trained, we can predict what background would have looked like, pure background, in the peak regions, right? So we can predict hypothetically what the background looks like. We can now regress out that background, right? Basically subtract that out of the observed ataxic signal in peak regions. Then we can fit this second model that learns to map sequence to the residual signal. That is the signal, the ataxic signal minus the predicted background, right? So let's see what that does. The hope is that this second model has now learned the cleaned sequence syntax that is devoid of the bias. So let's see if that works. So this is the same region I showed you before. Now let's see what the predicted background looks like from the bias model. Something quite shocking. You can see how similar the predicted background is to the actual ataxic signal in this peak region, right? Very similar, except around here. You notice how now when you interpret the background prediction through the background model, all of these spurious bases, they line up really well, but the CDCF motif does not. This makes sense, right? Because CDCF is driving chromatin accessibility. It doesn't drive background. The background model is trained on background regions, so it doesn't learn TF biology. So it can't really predict the CDCF motif driving activity. So you can see the differential signal between the actual ataxic signal in peak region and the predicted background is the CDCF motif. So now when we make the prediction of the residual signal, it's this beautiful footprint, this latent footprint that was hiding in this data that was corrupted by the TN5 bias. And now once we take this latent signal, the residual signal predicted by the model, and we interpret what is driving this background corrected signal, it's this beautiful CDCF motif, along with 2 other repressors, these ZBTV7As, okay? And this significantly outperforms any other approach for bias correction, ad hoc approaches of learning some KMA models or doing some position weight matrix models. They really don't work as well. Using a neural network to learn bias and correct bias is incredibly powerful. So you can take neural networks, Achilles heel, which is they learn whatever they see incredibly accurately. you can turn it on its head, make it learn bias very well, regress out the bias, and then use another neural network to really beautifully learn the underlying latent signal. So once you corrected this and you relearn the motifs from the bias factorized corrected model, all that TN5 bias is completely gone, and you just learn all of these beautiful motifs. Okay. So an interesting question is that DNA-seq and ATAC-seq, both of these assays, they both measure chromatin accessibility, but they're using different enzymes. So what do they look like? Well, let's look at this peak region. This is a smooth data from ATAC-seq. You can see kind of this nice peak. This is what the data looks like at base pair resolution. Now, if you train a Chrome BPNet model without any bias correction, it can exactly replicate the data as you see it. Beautiful prediction, right? Now let's look at DNA-seq for the same for the same cell type, for the same region. Again, you see this kind of beautiful smooth peak. So both experiments are capturing the same broadly accessible region. But at base pair resolution, look at the DNA-seq and compare it to the ATAC-seq. So different. Something is off, right? One of them is doing something weird, or they're all both doing something really weird. In fact, again, if you train a Chrome BPNet model without bias correction, on the DNA's data, it beautifully, recapitulates the DNA's data. So you can see a neural network will just sort of, learn whatever you tell it to learn, right? It doesn't really care. It doesn't really know biology. You give it data, it learns the data. It can make extremely accurate predictions. But there's clearly something off because both of these experiments are supposed to measure the same thing. They do kind of measure the same thing at a low resolution. At the peak level, if you smooth the data, they capture the same peak. But at base pair resolution, the profiles are extremely different. So now after bias correction, this is the bias corrected ATAC-seq profile predicted by the model. You can see it's dramatically different from the observed data. And you can see these kind of beautiful footprints, these two footprints that the model predicts. And this is the underlying sequence syntax that's driving activity, the 1, 2, 3 motifs. Now, if you use the same bias correction trained on DNA's data, you again see these kind of beautiful footprints with, again, the exact same 3 motifs driving activity. Now, they're not identical, and the reasons why they're not identical, the reason is because the TN5 enzyme is bigger than the DNA's enzyme. So actually, it not only captures the footprint of the transcription factor, but you can see there's an offset. The footprints are larger because TN5 kind of binds there next to the transcription factor once it tagments the chromatin. So its resolution of footprinting is slightly lower, but you can see that the footprints now align much better. And more importantly, the underlying sequence driver that the model's learning is nearly identical, right? That's great. And again, we can quantify this genome-wide. So this is the similarity between the observed profiles from DNA-seq and ATAC-seq experiments as measured. Y-axis is the Jensen-Shannon distance. Again, very high value indicates very poor concordance. Low value indicates very good concordance. You can see there's very poor concordance between DNA-seq and ATAC-seq at base pair resolution in the actual measured data. Once you do bias correction and you take the predictions of the model, so this dramatic improvement, right? So they get much more similar. More importantly, the underlying sequence drivers that the models learn from DNA-seq and ATAC-seq are quite different when you don't use any bias correction. After you do bias correction, they become very, very similar, right? So this is dramatic improvement in the concordance of this as well. And once again, we've trained using AnVIL and other resources, these kinds of models, Chrome BPNet models on over 3,000 DNA-seq and ATAC-seq datasets and single-cell ATAC-seq datasets, and these are all also incoming at the ENCODE portal within a month. Okay. So just in the interest of time, I will skip this portion, which goes a little deeper into a fetal atlas. And because I'd like to show you variant effect prediction, okay? And then I'll stop in about 3-4 minutes and give some time for questions. So an interesting application of these models is really predicting effects of genetic variants. Now, the models I showed you have not been trained on genetic variation at all. They train on the reference genome across the entire reference genome using a single representative experiment in some cell type of interest, right? So can these models actually predict effects of small changes like single nucleotide perturbations, even though they're not trained on any such data? They're just trained across the genome. So here's one example of what the model's predictions look like. We're taking a variant, which is QTL. It's a quantitative trait locus. So this variant it has been shown to have causal effects on binding of the SPY1 transcription factor. Here, the model is trained on chromatin accessibility. So it's not even trained on SPY1 ChIP-seq data. It's just trained on chromatin accessibility. And we take the reference sequence with the C allele. The model predicts low activity. We then swap the C to a G allele. We push it through the trained model. And the model predicts this beautiful, strong footprint, right? You can see this really dramatic amplification. And if you now interpret the model, you can explain why this activity increased. So you can see that the C allele, doesn't do much at this position. There's a weak SPY1 motif right here that's driving kind of weak activity at this site. When you swap the C to a G allele, it produces a very strong SPY1 motif. And this weak SPY1 motif and the strong SPY1 motif collaborate to produce this beautiful, strong footprint, right? So you get this very nice interpretation. Now, this is one example. Can we recapitulate big QTL experiments, right? So what are QTL experiments? What QTL experiments do, like in this case, we're looking at chromatin accessibility QTLs. They take a large panel of individuals, cell lines in this case, lymphoblastoid cell lines, from 100 individuals of African ancestry. We sequence their genomes, or we genotype them, and we measure chromatin accessibility in each of these individuals. So this is a very big, expensive data set, right? You have to perform ATAC-seq in hundreds of individuals, and you have to sequence their genomes, okay? That's the experiment. And then you will perform an association study where you're associating genetic variation at each SNP in the genome with nearby chromatin accessibility. And you're trying to find genetic variants that influence chromatin accessibility, like right here, right? Here's a genetic variant. You can see that individuals that have are homozygous for Gs have no activity at this position. But if they are heterozygous, that is, they have a T, or they're homozygous T, you can see they have activity, right? So clearly it's a genetically driven effect on chromatin accessibility. And when we perform this association study genome-wide, we learned 11,000 chromatin QTLs, that is genetic variants that drive differential chromatin accessibility in the same cell type across individuals. Now, we train a chrome BPNet model on a single reference example, an ATAC-seq experiment performed in a European LCL. So these QTLs were discovered in an African cohort. We're now switching ancestry. We're going to train this model on one ATAC-seq experiment in one individual on the reference genome. This is a an individual of European ancestry. And we will use this model in a counterfactual setting to try to predict the effect sizes of all of these variants. And this is what we see. We see very nice directional concordance, right? So this is the actual betas from the QTL experiments. These are the predicted log fold changes. So you can see, we see very nice directional concordance, and the Spearman and Pearson correlation are about 0.69. They're not perfect. Again, this is a local model. It's not able to learn long range effects and so forth. But even if you do this sort of QTL experiment twice, right, on two different cohorts, the correlation of these betas, the CAQTL beta, is about 0.8, okay? So we're not that far off. We're about 0.1. There's still a bunch to do, but it's remarkable that you have a model that's never been trained on any genetic variation. It's able to really get a pretty good correlation of effect size, and more importantly, get the directionality very accurate. So you can predict when a variant has a positive effect, when a negative effect, as I showed you in the previous example. And now we can contrast the performance of this small, simple model, this Chrome BPNet model that's trained on one data set in one individual, to much larger models. So here I'm going to contrast to Informer. Informer is a state-of-the-art model that was released in 2021 by Calico and DeepMind, by Ziga Abchek and David Kelly. And it's sort of the contrast version of our model. Our model is small. It's trained on 2 kilobase sequences tiled across the genome. It's trained on one experiment. You can train in 6 hours on a consumer GPU. Informer is a massive model. It looks at 200 kilobases of sequence at a time, so you can learn long-range effects. It is trained simultaneously on tens of thousands of ENCODE and roadmap data sets, and it requires massive TPUs to train this model, right? And even inference on that is extremely expensive and challenging. What's very interesting is if you now take these models, our Chrome BPNet model trained on one LCL, and you take Informa and look at its LCL heads prediction, you can see something very interesting. That is, you take Informa's published predictions of variant effects, and they do much worse. X-axis is different p-value thresholds for the CA-QTL, so different thresholds for calling significant CA-QTLs. Y-axis is the Pearson correlation, as shown right here. So the light blue and the dark blue are Chrome BPNet, and the yellow ones are Informa. There's a dramatic gap. So this was weird because, I mean, Informa is a much bigger model. We expected it to significantly outperform us, but something seemed off. So we went back into the model and tried to look into what the issue was. And we realized that they were predicting variant effects at 100 kilobase resolution. So if you have a variant having a small effect on a local peak, it completely gets swamped out, by looking at the profile at 100 kilobase resolution and looking at the allelic effect at 100 kilobase resolutions. This is a bug in their actual predictions that they released through their publication. But if you fix that bug, this is the green curve. So we can see that it still underperforms us. Even though we fix the bug and it's such a big model, it still does worse than us. This is just for quantitative prediction of QTL effect sizes. If you look at discriminating QTLs from background variants, even then, here's their prediction before correction. After correction, we are still very similar to them. And what's really interesting is that we simply boost the quality of the data we are training on. What I mean by that is we train on a deeper data set. So it's still our small little model. It's trained on one experiment, but we increase the number of reads in that experiment. So increase the quality of the data from 68 million reads to 572 million reads. You see this nice bump, right? So what this is showing is that you don't need a big model, actually, or at least a big model that's trained the way the current models are trained. A small model that's trained well on high quality data can outperform a giant model that's trained on lots and lots of data, right? And just to give you another contrastive example, there are these new DNA language models that have also been trained. Again, massive models, and they take days and weeks to train. Nucleotide transformer, Hyena DNA, Jenna LM, DNA BERT. And these DNA language models are supposed to be able to learn very powerful representations of DNA. And so we take these DNA language models and we actually train them on the exact same data that Chrome BPNet is trained on, right? But here's Chrome BPNet's performance again on two QTL data sets, just the ability to discriminate, QTLs from background variants. So you can see 0.9 and 0.8. And this is the performance of the 4 DNA language models trained on the exact same data, right? So whatever reason, they're actually not learning very good representations. They're pretty bad. And they can't even come close to what our models can do. It's why it's very important to actually, not just go with, scale is everything. How you train models, how you design them, how you sample the data, how you account for biases, and sort of systematic evaluation on counterfactual prediction tasks is very important to understand what these models are doing, okay? I will skip this in the interest of time. I'll just say that we can predict, variants within GWAS loci. Here's one really cool example. This is actually a coronary artery disease GWAS locus, and we train a model on mouse coronary smooth muscle cells. So this is a model that's trained on a different species on a very important cell type that's relevant to the disease, and the model beautifully fine maps this locus to these two variants that are both right next to each other, and they have beautiful haplotypic coordination. One variant on one haplotype creates a very strong activator motif and destroys a repressor motif. On the other haplotype, this other variant creates this very strong ZEB repressor motif and destroys the activator. So you see this kind of amazing switch in activity, complete silencing on one allele, on one haplotype, and dramatic activation on the other haplotype through this kind of coordinated effect of the two alleles. And this variant is in fact EQTL as well in many heart tissues, okay? So with that, I'll just summarize and say neural networks can, as I showed you, hopefully, they can very accurately model cell context specific regulatory profiles at base pair resolution. These models are black boxes in the sense that you cannot interpret their precise parameters, but you can actually use them as oracles to query them to understand all kinds of interesting biology. You can decipher motifs, complex syntax. I didn't show you, but you can actually distill thermodynamic affinities from these things. You can interpret these models to actually not only learn biology, but also learn artifacts in the assays. As I showed you, assay biases can be detected, learned, and corrected, thereby improving concordance between assays and actually revealing latent signal and biological drivers of activity. The models can be used to predict counterfactual effects of genetic variants. You can use them to fine map GWAS loci. We can also use them to study rare disease variants. I have some cool examples, didn't get time to show it. And an important lesson to learn is, at least for now, these small single task models can often outperform or perform on par with much larger multitask models. So at least for academics who don't have access to massive cloud compute budgets and huge amounts of local compute, these models actually can take us very far. Okay. And with that, I'll again thank all the students and postdocs and scientists in my lab, and all our collaborators and all our funding sources, including AnVIL for a very nice seed grant a few years ago that really helped us test and optimize training these models and allowing us to then massively scale up and train a lot of models for ENCODE. Thanks.
Thank you for an incredible presentation. Let's open up for a few questions.
Okay. Okay, I have a lot of questions, but I'll save it. So I'm really curious about the contribution of the reference to biases that you're observing right now. So obviously, all of the ENCODE data and so forth is all mapped to that same reference. Have you done any experiments to see how much bias that introduces versus mapping to the underlying haplotypes, right? Because presumably,
yes,
there is sequence variation that's inherently different between the reference and the samples from which you're measuring things, and presumably that does disrupt things at a certain frequency.
Absolutely. So what we see is if we train on a single sample, right, like this example, on the reference genome versus training it on the actual genome of that individual, we see almost no improvements at all. And the reason is because there isn't a ton of genetic variation that's causally driving effects, and the model is actually small enough that it prefers making mistakes. See, the thing is that model is learning across the entire genome, and it's trying to learn like regulatory syntax, right? It doesn't know about genetic variation. So it's actually quite immune to mistakes. It thinks of them as errors in the sequence. And so what interestingly happens is the model actually will make a prediction that doesn't agree with the observed data, right? So let's say the allele in the reference genome is incorrect. Then, let's say the allele in the reference genome was a G, and the active allele is a C, right, in the personal genome. In that case, the model will predict low signal, even though the observed data is showing high signal, right? So the model actually prefers making a mistake than overfitting to the observed data. Now, if you train these big models like the informer models I showed you before, they have so much capacity that they actually start overfitting to the reference. So the answer is a little complicated. If you have big models, it's actually quite dangerous to train on the reference because the models will often overfit to the reference. With small models that don't have a lot of capacity, they inherently regularize. They have to learn motifs accurately. And so if they see a wrong allele at a position, they would rather make a mistake than overfit. That being said, that's if you take the model and train it on a single sample and you train on reference versus personal genome, we don't see much difference. But there is evidence not from us, but from other folks, that if you have a decently sized cohort of individuals, like let's say 20 to 30 individuals, and you jointly train on all of them together, so you have a bigger cohort and you train on all of their personal genomes, you actually see improvements in variant effect prediction. You see kind of better, these correlations actually start improving quite a bit. So you don't see kind of directional discordance, but the accuracy of the actual effect size prediction starts improving, right? So that's the complicated answer. Hopefully that makes sense.
It does. Thank you.
Yeah, thank you for that. Let me take a couple questions. So I thought, you had presented really compelling data that the small model has a lot of advantages. At the same time, the data you're presenting right now says the small model is not perfect.
Yes.
I think you said, one of the characteristics that you're trying to model is that sometimes those long range interactions do matter and you may need a larger context window. Could you comment or speculate on, what sort of architecture, might be able to capture that? Is it just growing the window for a convolutional neural network, or is it some other architecture? I'd be interested to hear your thoughts.
Yeah. So that's a great question. So, we think that the Informer architecture, these transformer architectures are actually great. They're actually perfectly fine. It's not an architectural issue. It's the fact that the models are too large to be trained on the data that we have. So the issue with learning long-range interactions is that unlike local sequence models, right, where information density is pretty high in the local sequence and very similar across peak regions, right? As you increase the receptive field of the model, like in Informer's case, you're going to 200 kilobases, you have very sparse signal distributed in the genome, right? And you're trying to learn all of these kind of long-range interactions from a very vast diversity of spacings and sparsity across the genome. And there just isn't enough data to train it in sort of this one-shot manner directly from sequence to activity. So what we are thinking of doing, and to be honest, no one has really figured out how to get this to work extremely well. But what is very clear is that it doesn't matter how much you increase the size of the model and how many additional data sets you train it on, these models don't improve, right? As I showed you right here, it's not just Informer, but even the newer generation models like Borzoi and all of those, they have the same problems. They are unable to outperform even our small models by a decent margin, not just for chromatin accessibility, but I didn't get to show this here, but even for gene expression, actually chromBBNet trained on accessibility, actually outperforms Informer trained on expression itself, which is kind of shocking, right? And the main reason is because the long-range interactions are not being learned very effectively. So what we think needs to happen is we are thinking of building models hierarchically. So we don't try to do a one-shot learning of like a massive sequence to long-range effect profiles. We first want to learn very good local sequence models like chromBBNet, and then we piggyback off them. So these models have learned very good local representation of sequence. What they haven't learned is how one sequence, has this residual effect on another sequence, right? But if we decompose those tasks of learning local representations very well, and then only learning long-range interaction effects, we think the models will learn much better. Sort of, trying to help each phase of training focus on different aspects, right? Learn local representation very well, and then learn long-range effects. And also, we think that you need to provide the model some help. So just providing a massive sequence without any other information is like a needle in a haystack problem, and there just isn't enough data to learn. But you can do very simple things like Jesse Engritz has this very nice ABC model, which is a very good model. Like it's simply an activity by contact model. You can take the activity at a region, at an enhancer, and you can multiply it by the contact frequency. And just that multiplication is a very effective way of sort of capturing first order long range interactions. So what we provide these models like a scaffold, right, of approximately correct. They're not very accurate, these long range interactions, but they're still very good scaffolds. So you provide them some prior knowledge of prior scaffolds of what is expected to interact with what, and mostly have the models refine those interactions, right? We think those kinds of approaches will actually work very well. So this is still, no one's really figured out the secret sauce, but we definitely need long-range models to get to the next level. But what is very clear is that naively training these, just increasing model capacity and training on lots and lots of data sets doesn't cut it. There just isn't enough data to support an end-to-end training strategy.
Thank you for that. And in the interest of time, maybe just one last question, which is the same one I asked Taichel this morning. So you have our complete attention. So thank you for speaking at our conference and talking about AnVIL. If you had to name one thing that's been a challenge or a bottleneck or a need, how can we best help you? How could we best support the research you're trying to do?
I think AnVIL's been great, actually. It has really, in some sense, made things a lot more bulletproof. we had a horrible incident working on the cloud, a few years ago, not through AnVIL, but through regular GCP during the pandemic where, for whatever reason, we had, costs just run astray. And that's because, people can forget to turn off virtual machines and, rent a very, very large machine and, not care about how much it costs and so forth. I think the way AnVIL's designed as this sort of front end on top of the cloud that has a lot of interesting protections built in, I think is a real positive. I think the only thing I would say is it's not really an AnVIL issue, but I wish cloud providers had like a subscription model, like a fixed cost subscription model where, academics could say like, oh, I'm willing to rent these many, CPUs and GPUs for a whole year, right? And I can use it as much as I like, but just that much compute. And that's kind of like renting a cluster, right? And like you pay at your local academic installation. A cost structure like that, I think, would be even more beneficial. That's again not really an AnVIL problem, but if NIH could like negotiate some kind of fixed cost subscription model behind AnVIL, I think a lot of academics would switch to using the cloud through AnVIL than currently are, because they're still worried about these runaway costs that can happen when people work on the cloud. So to me, that's the only thing. Beyond that, I think AnVIL's been amazing. Like I think we really love the ability to automate things, how it's able to automatically pull resources, the Jupyter notebooks, all of those functionality is great. I think integration of the genome browser and so forth would be also very nice. And I think there are potentially ways to do that already through Jupyter notebooks with browsers that actually can operate within there. But those kinds of small things would make it even more powerful. But I think it's a great platform overall.
Okay, well, thank you for that. And thank you again for an excellent keynote presentation.
Thank you, and see you all at the tutorial.